So within recent years, nanotechnology has pretty much exploded, and it's found itself in various applications through many different industries. So obviously this field has huge potential, and that's why I'm super excited to be sharing with all of you watching this video a nanotechnology experiment done by the University of Berkeley. So what's interesting about this experiment is that the researchers actually use viruses as biological building blocks to create tunable nanomaterials. And before I get into the experiment, I just want to explain why it's so important. So the reason why it is so important is because of the functions of the nanomaterials that they created. These nanomaterials have the ability to be used as biosensors, piezoelectric powered circuits, and even in tissue repair, which is actually what I find one of the most exciting applications. And because of these applications and their potential to be developed further to be used in various industries like the medical industry and the electronics industry, that's what makes this experiment so important, because of the potential for the future. So I mentioned earlier that viruses are being used as biological building blocks for this experiment. And now the type of viruses that are being used are called bacteriophages. So just a bit of review on the bacteriophage. They're basically a virus that kind of looks like this. I'll try to draw it here to the best of my ability. So they're a virus that looks like this, and their function is to infect bacterial cells. And then by infecting these cells, they can actually use the cellular machinery to create copies of themselves, at the same time killing the cell. So the phage is actually divided into three main parts. I'll section them off right here. So the top is called the capsid, and really this is just a protein envelope that surrounds the phage's genetic material. It has this body, which is called a major coat, and it's really just a long rod of thousands of different proteins. And then it has the minor coat right here, which are these little tail fibers that look like legs. And this basically helps the phage move around and then land on certain cellular membranes to be able to begin its reproduction process. So this is the common type of bacteriophage. But what's interesting about this experiment is that this is not the bacteriophage that was used to create the nanomaterials. What was actually used is the M13 bacteriophage. And I'll draw it out for you here. It looks like something much different. So hopefully you can see from my drawing, but its structure is actually much different than that of a conventional bacteriophage. It still has its minor coats, but it actually has two, and then it has one large major coat as its body, and this contains its genetic information. Now, what's so special about this phage is that, one, its structure is completely different than that of a conventional bacteriophage. It still behaves in similar mannerisms and infects bacteria, but because of its different structure, scientists have found that you can more easily manipulate its genetic code or DNA sequences to be able to have it express different types of proteins. And this makes it really useful for modeling protein-to-protein interactions and understanding types of bonds. The other important key point with this bacteriophage is that it actually undergoes a chronic infection life cycle compared to the lactic or lysogenic life cycles of other bacteriophages. So now that you have this background knowledge, let's get into the experiment. So the experiment consists of four main parts, which I'll write out here. The first part is the creation of the genetically modified phage. Then the next is the amplification of the phage, and I'll explain what all these mean later. Then we purify it, and then we create the nanomaterial. So let's start with the first step, which is the genetic modification of the bacteriophage. So we have our M13 bacteriophage right here. And great, we have a normal bacteriophage. But the nanomaterials we want to create have a very specific function, and we want to be able to use these phages as the building block for those nanomaterials. So what we have to do to get them to bond to other phages in certain ways is to genetically modify them. And the way we usually do this is by inserting a certain strand of DNA, called a primer, into its genome. And what this does is that it actually changes its genetic sequence so that it'll actually express different proteins when the phage undergoes a replication cycle. So this is the genetic modification step of the phage. And now once we've created our genetically modified phage, the next step is to amplify it. And what this means is just basically use that phage and have it undergo its normal life cycle to create thousands or even millions of more copies of itself. And this is important because if we were to try to just create millions of copies of this genetically modified phage by genetically modifying every single phage, that would be hugely inefficient. So what we do is that we amplify it, we'll put it in a bacterial strain of E. coli, for example, because the M13 bacteriophage infects E. coli, and then automatically, without any interference from humans, it'll be able to create thousands of copies of itself. So then once we have our amplified phage colony, we then actually have a solution of phages in the E. coli strain. But we have a problem here. We have a lot of the bacteriophages that we want, which are genetically modified and have the functions that we desire, except they're in a bacterial colony of E. coli. So what we have to do is that we have to separate them or purify the solution to be able to extract the phages. So usually how this is done is that we have a machine called a centrifuge. And what it does is that it takes a couple samples, and we'll assume that these are the samples of our bacteriophages in the E. coli strain. Now, what the centrifuge does is that it basically takes the samples and then it spins them very, very fast. And because the E. coli and the phages have different densities, they actually are able to separate in the liquid because of centripetal force. So what we end up getting is we actually get a solution where we're able to extract just the phages by themselves. And then when we put this into a new solution, we have a pure solution of just M13 bacteriophages. And then now all it's left to do is extract the nanomaterial. So here is actually where the experiment gets really, really interesting and the most exciting. So there's many different ways to extract nanomaterials from our very pure phage solution, but the way that these researchers did it was actually quite fascinating. So what they did is that they used something called meniscus-based self-templating assembly, and that sounds like a huge term, but once you break it down, it's actually quite simple. So right here we have a very pure concentration of the genetically modified M13 bacteriophages. And we have millions of them all packed into this very concentrated solution. So what the researchers did is that they actually took a computer arm, or a robot arm. It was more like this metal stick or a metal strip, but then they put it in the phage solution. And what happens is that because these phages were introduced to a new material, they actually were able to attach themselves to this metal arm, creating a substrate in the solution. Now what's really interesting is that when this metal arm was pulled out, the phages actually were able to form the different nanomaterials by attaching themselves to this metal arm. But what the scientists and researchers noticed is that by changing the properties of the meniscus, and just a quick review, the meniscus is basically the point at which the liquid meets air, so right here. By changing the properties of the meniscus, they're able to change the type of nanomaterial created. So for example, let's just say this arm was being pulled out at 2 meters a second, then, and maybe at a 90-degree angle, then the nanomaterial created would be different than if this arm was being pulled out at a centimeter a second and at a 45-degree angle. So this way, very slowly. And this is really interesting because, and this actually happens because of the meniscus. So the meniscus has something called interfacial forces, which is basically the forces that determine the formation of the meniscus and how it interacts with other types of materials. So by manipulating those interfacial forces through the speed of pulling this metal arm out, as well as the angle, then the phages are able to self-assemble in different ways and create different types of nanomaterials. And I think this is a really revolutionary and interesting part of this experiment because it holds so much potential for the future of nanotechnology. So hopefully you enjoyed this video and you learned something new today, and if you did, that's great.